Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork perspective. So in this video we will talk about the Crassulacean acid metabolism. The metabolism mean the catabolism and anabolism. Anabolism mean the making some little molecule into a bigger molecule, and catabolism mean the bigger molecule convert into the little molecule for the using of as a energy. So here the Crassulacean acid metabolism is carbon dioxide fixation succulent plant in the succulent plant. So the succulent plant is the Crassulaceae. That's a Crassulaceae family that is known as CAM plant. That's a Crassulacean acid metabolism. So the CAM plant formed malic acid at night. So it means that malic acid produced and stored into the vacuole and disappear during daytime. That's a day and light. So that's disappearing. We will discuss about the whole mechanism. So first of all, we should learn the concept about the CAM plant. That's a Crassulacean acid metabolism. So the CAM plants are succulent plants. We will discuss about the here is a sample of plant which that is the reduced leaf. There's the surface to volume ratio reduced, and there's the reduced and stomata is low, and there's the succulent plants mean the vacuole is mostly filled with the water and something that's present in the very here is the cross section of the leaf, that is the spongy mesophyll cell is more and palisade mesophyll cell is not good developed. So that is the basic differentiation as compared to another plant. So here the bundle sheath cell, there's a red color shown in the bundle sheath cell, and the phloem is the brown color, is the fiber like structure, and xylem is the orange color which show there the xylem. And here the stomata which composed with the guard cell which used for the entry of carbon dioxide and oxygen releasing mechanism. So the light time, there's the too much low water in the ground, and there's the arid climate which that is the drought, and hot weather which that is not due to the respiration of water, because if water is respired more then the plant will be wilt. So here the mechanism of CAM plant, we will discuss about the stomata open at night, okay, and carbon dioxide enter into leaves. So it means the night time stomata will be open and absorb the carbon dioxide, and then here is the accumulation of malic acid in night time. So the stomata will close during daytime, and accumulation of malic acid in night time will be accumulated due to the absorption of carbon dioxide, and during daytime that's the disappear. That is the meaning of that things. So be concentrated and learn about the basic concept, and don't confuse about that, because it's very easy and simple. So here I will draw a bundle sheath cell which that is the here is the lower epidermis of the leaf which that is composed with the stomata, and here is the upper side is the mesophyll cell. That the mesophyll cell is basically present in the bundle sheath cells, which that is the near to the first of all we will discuss about the here this that is the basically is shown in the figure, that is the phosphoenol pyruvate are present in the mesophyll cell, which that is a carbon dioxide accept, and phosphoenol pyruvate is the 3 carbon, and that convert into the oxaloacetate, that's a 4 carbon by using of pep carboxylase enzyme. That's NADPH will be converted into the NADP plus, which that is the malate dehydrogenase enzyme is used to convert malate by the oxaloacetate. There's a malate is stored into the vacuole. So here is the vacuole, and there's the whole is the cell. Basically we will draw a mesophyll cell which that is the made up of sometime that's the bundle sheath cell basically. So here is another way is the night time, that is the malic acid store, and here we can check the sap of the leaf which that is the feeling the sour, that's the taste is sour due to the acidic. So that's the meaning of disappearing sugar and starch, disappearing the malic acid in the sap of plant. So that's why that's a vacuole during the daytime. Sorry, that's a daytime will be 4 carbon compound malate will be decarboxylation will be occur, and that's converted into pyruvate 3 carbon and 1 carbon in separate due to the decarboxylase enzyme which used for the carbon dioxide which enter into the which that is taking place in the cytoplasm, and thus enter into the chloroplast and use the Calvin cycle to produce 3 PGA. That is the 3 PGA, 3 phosphoglyceraldehyde. That's the 3 phosphoglyceraldehyde converted to the sugar. That's the sugar which use as a storage and starch and polysaccharide. That's the another thing which that is using for it. So here is the mitochondria, which that is pyruvate shown in the red color, which use into the mitochondria to produce ATP. That is the mechanism which that is the basic mechanism of that cam plant. So Crassulacean acid metabolism is the due to the Crassulaceae family of the plant which use that is the present in the arid climate, and that's a dry weather and something else. So here is the mitochondria which that is the pyruvate is used, and carbon dioxide not enter into it, and carbon dioxide will release from the malate to use in the Calvin cycle, and pyruvate is also used into the mitochondria to produce ATP as well as NADPH. So I hope you make understand about those things that is easy to understand. And now we'll talk about the basically the cross section of the cell of the mesophyll cell. So here is the carbon dioxide will enter sometime when night time, that's the enter into the into night time, that's the carbon dioxide fixation from here here to here. So here is the bundle sheath cells, and here is the lower epidermis of the leaf that is known as bundle sheath cell. So here is the revision with detail. We will discuss about the revise the whole mechanism with detail. So here is the cell which contain vacuole and mitochondria as well as chloroplast. That's the chloroplast used for the making during the light time produce ATP and NADPH, and Calvin cycle used to produce sugar, and phosphoenol pyruvate will be produced, that's go into the vacuole to store in the form of malic acid, and that's malic acid will go into the mitochondria by splitting of the carbon dioxide and pyruvate. That's the pyruvate is go into the mitochondria, and carbon dioxide will go into the chloroplast again to produce for a photosynthesis. So here the many species living in arid climate have thick leaves with relatively low surface to volume ratio and have low transpiration rate. Their cells have large central vacuole with a characteristic structure of these plants is that their stomata into basically organic acid. This help to minimize the water loss due to transpiration. So that's the family is Cactaceae, Orchidaceae, and Bromeliaceae and Liliaceae. That's the family which that is used to produce in the cam plant. Include these families in cam plant. So we will talk about the whole mechanism with the diagram. So that's I hope you understand with easy way. So here is the chloroplast, which that is I draw here is the chloroplast, and that's chloroplast is double membrane organelle. We use light and dark reaction and light reaction. That's a dark reaction is a Calvin cycle, and light reaction is the electron transport chain. That's the photosystem 1 and photosystem 2 is used for it. So here is the Calvin cycle is produced, and here's the mitochondria. In the other hand, there's the whole cell which that is I am describing it. So here is the mitochondria and its chloroplast, which that means the mitochondria is used for the pyruvate to consume and produce ATP and carbon dioxide because respiration. So that is why the carbon dioxide will be produced during the production of ATP. So that's remembering about that thing, which that mean the oxygen will be used to mitochondria basically and produce ATP and during the time of sugar and something which that is pyruvate in the add into it and produce ATP to as well as for using the oxygen also in the human being and plant. So that is the meaning of these things which carbon dioxide will be produced and Calvin cycle will begin produce 3-PGA, that is the sugar triose phosphate. That's triose phosphate is used for the ATP to make a AMP. That's a conversion of the basically that's conversion of phosphoenol pyruvate and that is a produced phosphoenol pyruvate. uh, that's that is the triose phosphate convert into the phosphoenol pyruvate plus when add the carbon dioxide, that's carbon dioxide which can be come from the mitochondria or can be come from the environment by entering of the stomata during night time. So that is the R reaction is occurring in the night time. So that's mean the carbon dioxide will be enter. So carbon dioxide will enter and the one enzyme which use phosphoenol pyruvate carboxylase, there's a pep carboxylate, there's a pep carboxylate enzyme used to make it. So that's convert the phosphoenol pyruvate by the exception of 3 carbon to convert 4 carbon oxaloacetate. That is the oxaloacetate is the acid and it's also known as oxaloacidic acetic acid. So that is the 4 carbon which that go into the NADP H using to convert NADP plus and there's NADPH used to give a proton to convert the malate. That the malic acid is also known as well. So malic acid is the 4 carbon compound, the 1 2 3 and the 4 carbon compound which that is the 4 carbon that is also known as the C4 cycle. So that's the C4 plant, it's also known as well. So because their product store into the vacuole. So C3 C4 plant is in the other hand we will discuss in the another video. So here is the just chem plant we are discussing about that. So forget about the C4 plant, you should think that and you should learn about the chem plant is here. So here is the vacuole which that is present and that's the vacuole, the channel are present with the malate ions, malic acid will enter 4 carbon acid by the entering of 2 proton by using of ATP, that is the active transport. And that's active transport will combine the proton and malic to convert the malic acid. That is the storing of the which that means the 2 proton is responsible for the storing of 1 malate. So the 1 malate is stored during the night time. So it means the night time that the taste will be sour due to the adding more malic acid by the production of malic acid, because malic acid not present before there's reaction in the chloroplast and mitochondria etc. But chloroplast is not, malate is present during daytime, because we will discuss about that thing, malate, how to stop malate during daytime. So here we will talk about the here the malate will be go outside and there's malate is accumulate into the cytoplasm and 2 hydrogen ion release and malate is inhibit the production of pep carboxylase. That's the inhibition due to the light. There's a light inhibit the not produce the malate and there's the stopping of the enzyme to activity to produce malate. So that is why that the malate will not produce and during daytime that's not malic acid produce and will not sap's taste is sour. So here is the malate which that is accumulate into the cytoplasm and go into the chloroplast, and then chloroplast will accept the malate and that malate go into the by the conversion of NADP reductase enzyme to use NADP convert into NADPH, that is the giving the proton for using it, and that's NADPH convert to the splitting of the basically pyruvate, that is the 3 carbon, and pyruvate is basically pyruvate will be produced, and that's the pyruvate. Here is the NADP reductase enzyme is used for it. So here is the malate will convert into the pyruvate by the releasing of one carbon dioxide, and that's go into the mitochondria, and this carbon dioxide we release to go into the Calvin cycle. So you should remember about that. Here is the Krebs cycle will begin in the mitochondria, and pyruvate will be used into Krebs cycle to produce ATP, and that's produce carbon dioxide, and carbon dioxide will go into the Calvin cycle to use to produce sugar. So that's very easy to understand about this thing, and if you not understand, so please comment below about that understanding, and comment about the artwork. So I will be thankful for you for watching this video. So here is basically here you should remember about that. So here is the carbon dioxide will be released. So carbon dioxide will be released also. So here is the pyruvate basically convert into the PEP, phosphoenol pyruvate, by the activity of ATP to convert AMP. So that's converted into the PEP, and this PEP is go into the as a phosphoenol pyruvate, as a use phosphoenol pyruvate, which that is also convert into this cycle. So that cycle will begin from here. So this cycle again will be PEP is produced from this cycle by the pyruvate. So here is very easy to understand. So thanks for watching, and comment below about that things, and subscribe the channel.